Good morning. This is Dr. Susan Love, and welcome to the Army of Women webcast on a research project by Dr. Kathleen Arcaro, using breast milk to understand breast cancer risk. I'm really very excited this morning to have this presentation because this was one of our early studies in the Army of Women that we recruited for, and we have some very interesting, if preliminary results. Dr. Arcaro is an expert in environmental toxicology, which is certainly an in topic these days, and she's at the University of Massachusetts. The research in her lab, besides the study we're going to talk about today, using breast milk as a marker of exposure of breast cancer risk, also involves looking at signaling pathways in estrogen receptor negative breast cancer, and also bioassays for the detection of endocrine disruptors in waterways. So she's very involved in the intersection of cancer and the environment, which is certainly a hot topic these days. The study that we recruited for in the Army that she's going to talk about today is looking at the cells normally found in breast milk. Certainly as the milk flows out, we shed a few cells with it, to see if there's any differences in those cells that could be reversed who are healthy. So she's looking at people who have had breast biopsies before, and then she looks at the breast cells and tries to evaluate them. And I'm very interested to hear today and hear what some of these results are. So with no further ado, I will have Dr. Arcaro start. And I've already warned her that if she gets too scientific, I'm going to break in and translate. And you're welcome to ask questions. If you think of something during the presentation, you can post your question, and at the end, we'll answer the question. Okay, Kathleen, all yours.
Thank you. Let me begin by saying thank you to the Army of Women and to Dr. Love for this opportunity to share some of our findings from the breast milk study. And I'm extremely grateful also to all the women who have responded so generously and donated milk at often very difficult times in their lives. So it's really a pleasure for me to be able to talk about the research that they have allowed me to do. So an overview of today's webinar. Today I want to explain to you why we study breast milk, what's special about it, and then why the study that we proposed really needed a different recruitment strategy, something really out of the box, and that will be the Army of Women. Then I'll tell you what we do with the breast milk, how we get it to the lab and what we do with it, and what we've learned so far in this study that we plan to continue because of really the fascinating results that it's providing for us. And we'll end by saying what we are planning on doing next and some things that we already have in progress. So the most...
Kathleen, can I interrupt for a minute? Can you try to talk closer to your microphone because it's coming through a little up and down.
Okay. How's it now?
That sounds better.
Okay. So this is really important to take home message already, is that the vast majority of all breast cancers are sporadic. And what that means is that the vast majority of all breast cancers are not due to inherited defects in the DNA. So most women with a family history of breast cancer never develop the disease. And most women who develop breast cancer do not have a family history of the disease. So this makes it really difficult for women to understand their individual risk of developing breast cancer. And the reason why we really want to understand an individual woman's risk of developing breast cancer is that we think we could reduce the incidence of mortalities. And this would be due to focused screening of women at high risk. You might get greater participation and acceptance of preventive measures. And it allows the women to have informed decision-making. So,
and we also might be able to figure out how to prevent it if we knew who was at risk.
Absolutely, and that's really my number two and my real goal, and I think what breast milk can help us do. It's prevention. So Unfortunately, most established risk factors do not accurately predict an individual woman's risk of developing breast cancer, and that's like being female, getting old, early menarche, late menopause, never giving birth, or giving birth at a later age. And also, most of these risk factors are not modifiable. So telling a woman that she has a risk factor and not being able to modify it, such as saying you have a family history of breast cancer, there's not a whole lot that you can do about that. So again, we are hoping that breast milk can help. With molecular biomarkers in breast milk may provide an answer. And just as the molecular biomarkers are helping us define breast cancer subtypes and then guiding treatment, there's many researchers think that there are molecular markers that can identify individual women who are at increased risk. And then, as was just mentioned, hopefully lead towards prevention.
So by molecular markers, you mean things that are changed in the DNA?
Absolutely. It could be changed in the DNA, or it could be changed just a protein, but that would probably come also from a change in the DNA or some other signaling pathway. So I do mean DNA, and that's what I'm going to tell you. But I think protein, other changes would fit under that molecular biomarker category. So what do we need of a molecular biomarker? We need it to be accurate, right? You can't have something that's not specific and we need, or not precise. We need it to be non-invasive. We really don't want to have to be giving women biopsies every year to check for molecular biomarkers. And this third one is really important. It has to play a role in the etiology of the cancer, ideally. So not only to tell us that the marker is a marker of risk, but that the marker, if we could reverse that marker, we would reverse risk. And that's really
so
the
etiology means the cause.
The cause, yeah.
Right.
The cause. And it needs to occur very early in the development of disease to be an ideal marker, right? Very early so that we can catch the disease before it's a cancer. And most importantly, we really want it to be modifiable. So that's a lot to ask of a biomarker, but that is what we ask and what we hope that we can get out of breast milk.
So an example would be HPV and presence of HPV and cancer of the cervix.
Absolutely, yeah.
It's involved in the cause. You can find it fairly non-invasively. It occurs early in the development, and we can change it.
And you can treat it. That's exactly what you want, right? So one of the things then that the way that we're going to approach this is looking at epigenetics. And epigenetics means above the DNA, so it's modifications to the DNA that are not mutations. And that's really important because mutations, we haven't really figured out how to correct mutations. But if you could think of little changes to the DNA or dressing to the DNA, we do have some clues as to how to reverse that. And many people think cancer is as much an epigenetic disease as it is a genetic one. And we already know that our epigenetics, so it's sort of the dressing of the DNA, changes as we get old, and it changes with disease. So the focus of our research with the breast milk is to look at epigenetic changes. Now, in my next slide, I'm going to give you a little one-slide overview of the epigenetic change that we're interested in. So there are several types of these changes to the DNA, again, that are not mutation, but what's also important about this change is that when it occurs and the cell divides, the next cell gets the same change. So DNA methylation is one type of epigenetic change, and it's basically the addition of a carbon and 3 hydrogens, so a molecule, to the promoter to a specific region in the DNA. And what this does then, if you can look here at what would be a strip of DNA in a normal cell, in a healthy cell, there's no little methyl groups here up in this promoter region. And that means that this cell then can produce the RNA and make a protein that might be important in repairing DNA or controlling cell growth or eliminating toxic compounds from the cell. So it's normal, healthy cell function. But these carbon and hydrogen, the methyl groups, get added in disease or in pre-disease, And you see it here, and then the gene is turned off, and so the RNA and protein are not made. And this is what we study in the milk cells from the women that we get. And when you look over
one
here,
way I think about, in my simplistic surgical view of methylation, is that if you think about say a light switch, if you totally destroy the switch, you can't turn the light off and on anymore. That's a permanent mutation.
right.
But if you put tape on the switch, you might have trouble turning, you might, but you couldn't turn the light off and on, but you could take the tape off and then it would be able to do it at work again.
I think that's a perfect description of the epigenetic or the methylation, yes. So when we say that methylation is reversible, really we're saying let's take off the tape. Let's take off
Correct.
the tape and turn the gene on or turn it off, with whichever is needed to do. So indeed, many researchers agree that DNA methylation patterns are one of the most promising biomarkers for assessing breast cancer risk and any cancer risk. And while most researchers agree that this is true, obtaining the tissue from healthy women presents a challenge. So how are we going to keep looking at their breasts to see if it's methylated? And basically the answer is we just need the cells from the breast. So one way of getting these cells from the breast, and the way that we've chosen, is breast milk. And so there are millions of these cells naturally peel off the exfoliated epithelial cells, and they're present in the milk. They represent the entire breast tissue, right? They're coming from all the ducts. They're easy to obtain. Women are just incredible in their willingness to donate breast milk. And importantly, you could do some repeated sampling, and that's helping us figure out a lot of basic epigenetic biology by sampling women when they have multiple children or sampling them through time. And there's one other really important aspect of this, is that every woman can be her own control because you can sample both breasts. So the study we wanted to do, we had some preliminary data that really showed, yes, you can look at these epigenetic signals in young women, and we collected milk from about 100 local women, and we saw these signals. And what we really wanted to know is, well, do they just represent, tell us that, yeah, this milk came from a woman who's lactating, or are they telling us something about risk? And so thinking about how we could get milk samples from a population of women who, at least a small subset of them, would be at increased risk, we realized that we needed to go national. And we proposed to study 250 women who have had or were scheduled to have a breast biopsy. And roughly, we knew that a very small percent, but a certain percent of these women would go on to have cancer, likely, and most of them would not. So we wanted to collect these women from across the country. This is how we do the study. We send her a box, and she fills it out. We obtain her biopsy reports and questionnaire, and we're going to do long-term follow-up. But the problem is no one believed that we could get these women. Women who are nursing and have a breast biopsy, a need for a breast biopsy, are a tiny population. And that's when we turned to the Love Avon Army of Women. And it just changed all of our recruitment problems. So how do we recruit? Conventional recruitment, we think of it as we went through lactation counselors, WIC coordinators. We sent emails to probably way over 1,000 people now. We actually contacted breast imaging centers. We thought that we could get these people to collaborate, and if any lactating woman came in and needing a biopsy, that they would contact us. We went to the OB/GYN offices and professional organizations. But we decided the web-based recruitment strategy would be a better approach for us. We did a whole lot of things back then, listservs, message boards, and tried some paid advertisements and a lot of other websites, and we really were down in the single digits of collecting breast milk. And then with the Army of Women, just 2 e-blasts gave us over 233 contacts. So these were nursing women who were willing to send us their milk, and they were either scheduled to have a breast biopsy or they had one, and they were also willing to send a breast biopsy report.
So just a great example of how efficient and how we can accelerate research through the Army of Women.
Yeah, it is interesting. For many people thought this was a study that should be done, and every grant reviewer said it can't be done because you won't find the population. You won't be able to get the women. If a woman is scheduled to have a breast biopsy, she's going to be too busy with other things and she won't send her milk. But that's not our experience with the women who register for the AOW. So how has the AOW recruitment helped us? It really was a huge success. Almost all of our participants, well, over 80%, come from the AOW. Some of them who may not come from the AOW directly may come indirectly from the AOW, because we have women contact us who say, oh, a friend heard about this study and she lives in Colorado and I live in Pennsylvania and I'd like to donate milk. So it's a little bit unknown how many come. After each e-blast, we receive these frequent updates with a list of interested participants. And that's really important, because I think for some of your other studies, you might wait and send an e-blast when you have a certain number of women. But when we contacted the AOW and said, no, we need frequent e-blasts, like every few days. When a woman says she wants to send us a milk sample, we want to know immediately. Well, the AOW modified their procedure to get us that information in a really timely fashion. And our recruitment coordinator then contacts each woman by email. So almost all of our research is done by email. We would do regular mail or phone, but that hasn't been used heavily. And importantly, most of the contacts that we've gotten from the AOW were eligible and chose to participate. So we're way over our 250.
That's great.
Yeah. So,
and quickly.
Yeah, and very quickly. So here's how it goes. The milk samples, we ship out this black box here that you see on your right. And in the front, the woman puts her fresh milk. And this is really important, that for our research, and I'll show you in a minute, we really do need the fresh milk. So the woman puts her fresh milk, left breast, right breast, and in the back she has these bottles, and she can put some frozen milk if she has any, or any extra milk. And we have other studies that we can do with the frozen milk. And this is an ice pack that she puts in the freezer. We try, and then puts it back in. We try to make it easy for the woman, that the box just closes down and latches, and she calls the express mail delivery, and they pick it up and get it back to our lab in very quickly. So this year we had participating women reside in 36 different states. This is for one study that we just published this past December in Epigenetics, and this was an analysis of 134 women with benign non-proliferative lesions. So you can see it's a totally different.
By that you mean they had benign lesions, but they weren't precancerous or didn't look in any way high risk.
Correct. That's from from their biopsy reports. Yeah. And so as you see, like for anybody who does breast cancer research, it's really unusual right away to have your women coming from 36 different states. So it's really a different type of research, not going through doctors' offices or hospitals, but research being done directly with the women who are providing the milk. So what do we do with the milk when we get it? The first thing that, and again, we need to have the fresh milk, because we want to take out, there are many different cell types, and we want to separate them. The cell types that would become cancerous, if cancer were there, are these epithelial cells, and we can pull them out of the milk with antibodies. And that's what we do. We use an antibody system, and we separate out the two cell types. And so that's important to us, that we have this population of enriched and depleted, or population of epithelial cells and non-epithelial cells. And then we go through this process that allows us, after PCR, to sequence and to tell us how many of the potential methylation sites are methylated in genes that are known to get methylated in breast cancer. So we focus it on target genes that get methylated in breast cancer, and when they are methylated, turn off proteins that are important for normal function.
And epithelial cells are the cells that line the milk ducts, and all breast cancer starts in those lining cells. So just to make sure everybody's on the same page.
Right, and that's important. I show a little bit of data later from the epithelial cells and the non-epithelial. And the non-epithelial would be blood cells and macrophages, all the other cells that normally would be present, and you get very different results. So that really is important. And we do archive the milk, and a few women have commented there's an option if they don't want their milk archived, and then we just toss out the rest of the milk. But for women,
by archived, you mean you store it, you freeze it and store it for other studies.
Right. So, and in fact, the women give us permission to retain the milk, to store it, and to use it in other studies that will help us understand breast cancer less. So it would only be for a study that was related to breast cancer, and it would be something like maybe trying to understand if levels of bisphenol A in the milk might cause a problem. So it's all related to breast cancer. So I am going to show some data, and I just wanted to show how I will present the data. This is called a box and whisker plot, and this is the median score, the score that occurred in the absolute middle. And we have the top, the percentile, the top of the box represents where the 75th highest score would have occurred, and the bottom of the box where the 25th lowest score would have occurred. The important point for this, these whiskers here tell us something about the range, right? So this is 1.5 times that range. But the important point for us is these outliers. So each outlier represents an individual woman's score. And the reason why we're interested in outliers is because most of the women who provide milk samples with us, their methylation scores should be low. Even though they've had a biopsy, their risk for breast cancer is really not that high above average. But for a small percentage of women, their risk may be above average. And we're trying to find out who those women are, to identify those women so that in future they could receive preventive care. So let's take a look at some data. All right, here's the data, probably the big take-home message. So I'm going to go through this and spend a little time on it. Over here you have RASF1, SFRP1, and GSTP1. Not important acronyms other than to understand that these are genes. These are names of genes that are important in normal cell function that get methylated in breast cancer, and when they're methylated, they're turned off, so there's no protein made. And the goal of this project here was to compare two groups of women. One we call a reference group. This is the group of women who had not had a biopsy, were not in any way at increased risk, and then we had our biopsy group here. Both groups are very similar, right? They're both lactating women. We check that they're of similar age and reproductive history. And the thing that differs between them is their risk. And what's really, really important to notice is that most of the women, so these are individual CPG sites, they're going across here, and that's the average. Again, individual CPG sites, 13 CPG sites on the average. Most of the women, the median scores, right, these average scores, are not different between the two populations, or not very different. So most of the women, regardless of which population they're in, they're not highly methylated. It's these outliers here, right, and there's some in each, but more over here, that will tell us that, yes, in this group that we would think is at slightly higher risk, there are more women with outlier scores. And it's these outlier scores that we think are indicative of increased risk. And these are the women, we want to follow all the women, but these are the women who potentially, if our hypothesis is correct, could truly benefit from intervention to reduce methylation.
But
Any questions
there? There's a couple
there?
of people who are outliers, at least for the RAS1 and the SFRP1 in the reference group too.
That's correct, and you would expect that of 100 women, there will be 1 or 2 who will be at increased risk. What's really important, if you look over here, when I say that the women in the biopsy group have had at least one breast biopsy and 30% reported a mother or sister with breast cancer, well, we know very well that those 30% are not all going to have breast cancer, right? So you can see right away how just having a mother or a sister is not enough to know what your risk is, and we're hoping that through long-term follow-up we would be able to say, yeah, methylation either is or isn't, but if it is, then able to more accurately predict risk than women who have a mother and sister with breast cancer, but in the future learned, oh no, my DNA is not methylated. They would have peace of mind because they would know that, okay, I come from a family with increased risk, or I have had some breast cancer in my family, but I myself am looking pretty good. And I think that's as important as telling the women who have high risk, high methylation, that they really need to do something to reduce that methylation.
Yeah, and the fact that in the reference group, I just want to point out that those women, that's all also breast milk, and so everybody's breastfeeding. So it's not the fact that just because you're breastfeeding, your cells might be more struggled, more abnormal, but it's that when you compare everything else being the same, the group that had a previous biopsy, more people were out of the normal than not. So that's how I'm just saying in terms of how you do research in people or in animals, it's really important to have everything else that you can control be the same, and only one thing that you're looking at have the chance of being different.
Yeah, I think that's incredibly important, and you can't stress it too much, that if we didn't have the reference group population there that were breastfeeding women of the same age and reproductive history, we really wouldn't know if the methylation on the left-hand side of the biopsy group wasn't just caused because the women were lactating, and maybe lactating breasts are different in methylation than non-lactating breasts. So,
right.
So that's very important. And in the next slide, this is a little different comparison, but these are the same women in each. This is just a single tumor suppressor gene, it's called. These are women with all different breast biopsy categories, so not just. But what we're looking at here is for the same women, their biopsy breast over here there are more outliers than in the non-biopsy breast. And that, I think, is important. We would have predicted that, but as you can clearly see, there are a few cases where the non-biopsy breast, would represent a single individual for these sites, is high, and that's important information. And so, again, I think it's demonstrating that the breast milk is really useful and that it's easy to look at both breasts, which is important, not just the breast that received the biopsy. And it's turning out to be useful in that we are finding specific signals associated with a biopsy breast.
So there are some people, it may be that they're at increased risk because of some genetic or some other exposure that both breasts have seen. Absolutely. And then there's other people where it's one breast that looks like it may be at higher risk. Maybe biopsies increase methylation, or I say this with trepidation because I'm a surgeon, but maybe surgery causes methylation. What about that?
Well, I think that's a great question and a great thought, I don't have enough data to address it, but I do have some. And some of these women gave us milk before their biopsy and some after. And when we have more data, we will be able to look at that. The other thing we do have is some women who gave us milk even though they had a biopsy 15 years prior. So if once we have more data, we'd be able to examine the relationship between timing of the biopsy and the milk donation. But we really don't have enough yet. That would probably take closer to 1,000, and we're still down in the hundreds.
Okay. Yeah. Well, it'll be interesting to see.
Yeah. And one final slide from this group is just that, again, our numbers are too small. But as you would expect for women who did have cancer, who were diagnosed with cancer after they gave us a milk sample, their biopsy breast for, again, a single gene was higher than their non-biopsy breast. You would expect that. But these numbers are pretty small. This is a sample of 4. So 4 women only included in this.
Big graph, small numbers.
Right. But there are other ways, and I think this is important, a few other things that we can do with breast milk. And here's a case. If you think about it, each woman provides an opportunity for us to look at DNA methylation in 4 different samples of her DNA. So she gives us a sample from her biopsied breast and her non-biopsied, so that's 2. And then each one, because we separate out the cell type, we look at both the epithelial and the non-epithelial, all the other cells. And so this graph here shows a woman who had a complex fibroadenoma, and you see that the only methylation that's high, this is for a gene called RASF1, promoter gene, a tumor suppressor gene again, and these are individual CPG sites. And her methylation is high at all these sites, but only in the epithelial cell population in her biopsied breast. The other two, her non-epithelial, the leukocytes and all, are quite low, and this is the non-biopsied breast, the epithelial cell fraction.
And I just want to point out that a complex fibroadenoma is completely benign. It's a benign lump that's not uncommon in young women.
Right. So we may learn, and this is really important, and I'm so glad that you point that out. We may learn that methylation is not specific enough, or methylation of RASF1, this target gene, is not enough to tell us anything about risk, because here we're saying it's methylated, but this is a benign situation. So it may be, and other studies are showing, that you really may need to look at a panel of 12 or 15 genes before you can give accurate, precise information about breast cancer risk. And here I want to show another. Here's a woman who had an angiolipoma, so you'll be able to comment on this. Not at all epithelial cells, not an epithelial cell. And interestingly, when you look, here's her biopsied and her non-biopsied breast. So a sample from each breast is high in methylation, but it's the epithelial depleted. So it's her blood cells that are showing high methylation, where here her breast cells are extremely low. So again,
an angiolipoma is a fatty tumor that has some blood vessels in it. So it sounds fancier than it actually is.
Right. Right. But it is interesting that her breast milk actually correlated very well with her diagnosis that there's no increased methylation in any of her epithelial cell fractions.
Right.
Okay. And then when starting to thinking about prevention is really where we want to go, and actually how we got into this. And we initially we wondered if having a child at an early age is thought to reduce your risk of developing breast cancer. And we wondered if maybe having a child at an early age, if it didn't reset your methylation clock. So the next graph is really just a case study, only 2 women, no data to really write home about, other than that it is interesting and suggests that we do need to study this further. So here we have 2 women, participant 2 and participant 4. Down here we have 1, 2, 3, 4, 5 tumor suppressor genes, and we're looking at the mean methylation overall. And so you can see that the participant who had a child at a very early age and had multiple births has very low methylation, whereas a woman who has a child at a later age and only 1 birth happen to have high methylation. Now, I selected these 2 just to represent the extreme differences between this. I don't have enough women who had children at early age and enough women who had children at a lower age to clearly analyze this. But if methylation is indicative of risk, and if we can see an early effect of pregnancy and lactation on this, we may be able then to say, okay, we know that early pregnancy and lactation reduces risk. Can we then reduce the risk for all women who having an early pregnancy is clearly not a good solution, but possibly reduce the methylation of those women who have high methylation by some other totally non-invasive method. And that's what I show on the next one, by diet. And I think this is really the future that this breast milk study is allowing us to go. So if you think that methylation is a problem, is important in breast cancer risk, then we need to change it. And there's evidence to indicate that sulforaphane, the compound in broccoli sprouts, really can alter methylation. And in fact, there are 2 clinical trials for breast cancer, for women with breast cancer, ongoing. But what we're proposing is a study of totally healthy women who are nursing and who want to eat more broccoli to reduce the DNA methylation. And this would provide an opportunity to look at repeated sampling, which is very difficult in almost every other type of study, to get repeated samples over a long period of time, so women who might nurse for as much as a year. And the other thing that is really preliminary, but there are a few labs who are suggesting that stem cells, epithelial stem cells, are present in breast milk. And if that's the case, examining the pattern, the methylation pattern in these stem cells, where many researchers think that stem cells might be critically important in the development of breast cancer. So therefore, examining the methylation pattern in these stem cells could be extremely important in seeing if we can change that methylation pattern. And then I also want to tell you that there's one other study that is ongoing, and the AOW is, again, helping with this one. And that's because many of the women, the majority of women in our study, were Caucasian, and we want to increase the representation of African American women in our research. And so the Avon Foundation has funded a project to recruit 200 African American women to donate breast milk. We're also trying to get more African American women to join the AOW, and we're working both nationally and also locally. And that's really important, because one question people do ask is, well, do you think that there's just a difference in the women who participate in the AOW, and you're going to find different methylation patterns there than all the other women? And so here we will be able to compare again the among African American women, the recruitment through the AOW, and the local recruitment. Even though we don't think that's a problem, we think controlling with the left and right breast is sufficient. But this is our next study, and we are already recruiting with the AOW. And then two last slides. I really want to thank the people in my lab, my collaborators who do this work, and the funding, the Army of Women, and the Avon Foundation. But specifically, my thank you also goes really to the women who have just been incredible at donating milk, sometimes just hours after finding out that they have breast cancer. So with that, I think I'm ready to take questions.
Well, thank you, Dr. Arcaro. And so if anybody has questions, now's your time. We have a couple, and I'm going to wait for you to put a few more in. I want to ask a question. There's some very interesting work from Pepper Shadeen that you're probably aware of. She's in Colorado, and she's shown, at least in animal models, that during pregnancy and lactation, and for the next 5 years, you have a higher risk of breast cancer. And then having been pregnant, you have a lower risk. And one of the questions, one of the interesting things is at the end of lactation, when you have involution, all the ducts and ductal tissue dies off, and then you make new ones. And so I wonder if that resets the methylation. So when you finish breastfeeding and you have involution, you kill off all those methylated cells and you start over again.
Yeah.
Think?
You know, that is such a perfect question, because one of the, I would say, I do know Pepper Shadeen's work, and it's really quite fascinating. One of the things with our study is that these women have donated breast milk at all times, from 16 days after birth all the way through 2 years and over 2 years. Some of them are still nursing. And so it really, we really need to look at that. We need to look at the methylation early on in lactation, the colostrum, the full milk, and then get those samples that are right there at weaning. We are now going back.
Well, we could also get ductal fluid when they're finished lactating.
That would be ideal, yeah. And we do have to go back and ask, in case any of those women are listening, we plan to go back and ask, when did you wean? And to see when how close our milk sample was to the weaning. But you're right, it'd be great also to be able to look at the cells in the breast after the woman has weaned. Yeah. And to also to see clearly, did length of nursing have any effect? Yeah.
Yeah. So there's one question here from a participant, asking if, one, if we're going to share individual results, and the answer to that is no, because even though this is very interesting, we don't really know what it means yet. These are preliminary results, and so it would be a big leap to say you have this methylation and therefore you're at higher risk. We're still trying to figure that out. So we are not going to, we will not share individual results because we don't know what it means. And she also asked if there's a Facebook page for this study. That I don't know. Kathleen?
Facebook. There's a website, and I think we're getting up a Facebook. I'm sort of slow on the Facebook, but if she wants to send me an email, I will check and see if the Facebook is up. But you do see our website there, and actually, as soon as we get a Facebook, it will be up on our website.
Liz asked, she said her son nursed on her cancerous breast for 10 months. Is there any evidence that this was harmful to him in any way? And the answer, I can answer that, which is no.
Yeah.
It's not harmful to him. Those cells are digested in his GI tract, just like, you know, cereal and everything else is digested. So you can't pass on cancer through your breast milk.
Correct. Yeah. I do think
so she shouldn't worry about that.
Yeah.
No. Another question from Mark is there? Do you think a modifiable marker for breast cancer could also be something like high cholesterol in breast milk, sort of like the HPV example that I mentioned?
Like high cholesterol, I think there could be a protein in breast milk. That would be easier. Maybe not. Yeah, I do think that there's that's a possibility. I am collaborating with a few people, and that's another benefit of this breast milk, is that there's enough resource there to collaborate. So I have a collaboration just beginning to look at different glycosylation of proteins in the breast milk. Someone else is interested in looking at potentially infectious agents in breast milk. So certainly that would be wonderful if there were some protein or other substance in there that could really be used as a marker of increased risk. And then we
actually have a large study we completed in China of 1,000 women where we collected breast fluid. This is non-lactating women, and looking for a marker, 3 markers, potential markers for risk, and found out of the 1,000 non-high-risk women, 4 women who had these protein markers that you could do on a sort of in a home test, like a pregnancy test. So we're refining it, and then we're going to do it in the Army of Women and track women over time and see if we can predict it. Because the one limitation of breast milk is not everybody pregnant or breastfed. So if we could do something similar on breast fluid that everybody has, then that might be easier.
I think so. I think the benefit potential of breast milk is that you get so much of it. So if the results are generalizable to the total population, what we can do is use breast milk to learn. And
then try it. And, yeah.
Exactly. It can guide some of the studies that we do when we can only get a drop or 2 of fluid. Yeah. Yeah.
So here's a question from Shannon. Oops, my screen just in the middle of my question. Here we go. Is there any suspicion that women who have children early and regularly are literally flushing cancer cells out during lactation over the years? Or, you know, we could say flushing the methylated cells out.
So I don't strictly know the answer to that. There have been hypotheses that you would be flushing out pollutants potentially, and you would be flushing out,
I mean, there's a lot of flow, so nothing's sitting there very, very long.
Right. So
another more research to figure out.
Yeah.
Natasha wants to know if there's any ethnic differences in the sample data collection results of the study, and whether there's similar studies in Southern California. This, I should say, Natasha, this is a nationwide study. You can be anywhere, so people in Southern California certainly can participate. for non-breastfeeding women, we at the Dr. Susan Love Research Foundation in Santa Monica are doing studies on breast fluid and looking for volunteers for that. So here's the picture of the national map. So you can be anywhere for this study. If you're breastfeeding, and if you're not breastfeeding and you're in Southern California, give us a call because we have a study for you. In terms of the ethnic differences, Kathleen, I gather that's why you're trying to get more African American women in your study.
Absolutely. It's too small to say. The vast majority of our women are Caucasian. We did try to analyze it, and there are no differences. But I think we will be able to speak to that soon. And there's good reason to think that there could be differences. The disease differs somewhat between African American and Caucasian women, and we need to see. We need to look at it. So it is what we'll be doing over the next year.
So I've asked the questions we have up there. If you have any other questions, speak or type now, I should say, rather than speak now. And up here comes one from Debbie. Is it accurate that Hamlet in breast milk can actually kill breast cancer cells in the ducts? Currently pregnant with breast cancer and hoping breastfeeding will help tumor shrink. I have some knowledge about Hamlet if you don't.
Go ahead.
Kathleen.
Mm-hmm. Yeah, no, please go ahead. Yeah.
Hamlet is a protein that's made in breast milk that actually does seem to kill cancer cells. It's the work of a researcher named from the Lund Foundation in Sweden, and they're trying to see whether it has value outside of breastfeeding. So there is data, but there's not a lot of data in people about Hamlet. And it's hard to know, Debbie, what's going to happen in terms of whether the breastfeeding will help the tumor shrink or not. The question is whether the tumor has destroyed the ducts and the lactating part, so maybe will not, produce the Hamlet. But it would be interested to find out what happens, and I think it could give us some good insight into what's going on. Most of the work in Hamlet has been done in rats and mice. Here's another question. When will the Army start recruiting for the African American study? I think we are already recruiting for the African American study. So if you go on the Army of Women website to current projects, you will see the milk study is the title. Maybe we should say, and that's the one where we're specifically looking for African Americans, Latinas, Asian women, and other ethnic minorities to be part of this research. So we are currently recruiting. You can go on the site right now and RSVP for this study if you would like. So go to armyofwomen.org, then look at the area that's called current projects, and you will see the milk study, and you can sign up. And then if you know anybody, or if any of you know people who are breastfeeding, particularly African American women who've had a biopsy, please pass this along.
With that, I'll just take a moment to do a little technical thing here. So we have several studies ongoing, and for the biopsy study, you're absolutely correct that we are recruiting for African American women, and our internal review board approval allows us to do that. We were just funded by Avon for another study of African American women who don't need to have a breast biopsy, and that study we're still waiting for our IRB approval, and you could appreciate that there are little subtle differences in the consent forms and all, and we should have that any time. So there may be a few women out there who contacted us and who said, we said, oh, we have to wait a week or so. But if you are African American and you have had a breast biopsy, and that's the consent form we would send you, then yes, we can accept that breast milk today. So you could go on the site and fill that out.
Okay, if there are no other questions, I want to thank Kathleen Arcaro very much for this great presentation. This webinar will be put up on the website, so if you have friends or family or know other people who couldn't attend, but are interested in the topic, they can come look at it at any time. And we thank all the participants. One of our big goals is to close the loop, and particularly when you're recruiting people for studies from all over the country, it's sometimes hard to get the results to them. So we're very pleased to have this opportunity through the internet to be able to close the loop and give you some of the results. And as more of the results come in, we will follow up and give you more results.
Okay. Thank you very much.
Thank you very much, Dr. Arcaro, and good luck with your further research.
Thank you. It's really been terrific. Thanks.